So, 1803 and another Neuro Talk Evening, number 11 for me. Today's guest is Professor Andreas Keil. Good to see you. He's from the University of Florida, and today's topic is the building blocks of mental health, linking basic science of brain and behavior to emotional well-being and resilience. And just to introduce you a little bit to him as a person, it should switch now, but sadly it isn't actually. Ah, there it is. Wow. So he is a distinguished professor of psychology. As mentioned, he's currently at the University of Florida. He has an interdisciplinary background because he also holds a diploma in mathematical statistics as well as psychology. And he uh, received his PhD in psychology from the University of Constance while focusing on oscillatory brain dynamics. He has previously been the professor for clinical psychology and cognitive neuroscience at the University of Constance and was a visiting researcher at the NIM Center for the Study of Emotion and Attention. His research mainly is about, but it's really hard to say in his case because, as he told me, once a while, every around about 5 years, he's completely doing or starting a new topic, and that's what he likes about science. So at the moment, temporal dynamics of emotion and attention using EEG and steady state responses, so steady state visual evoked potentials in this case. Notable works a notable work is frequency tagging and methodological pioneer based in competition models. Um, Current studies also focus on the sensory plasticity and field learning, so the correlation between, so how do we adapt in terms of field learning, how does our brain adapt, and what changes in terms of perception. And with that, we will come to today's talk. Then many treatments at the moment focus on top-down regulation. So how can we change the behavior of a person if there is something like an anxiety disorder? How can one face the stressors that are surrounding them? But we are often neglecting how bottom-up perception is changed at the point of an anxiety disorder that already shows. So how can we integrate both of these processes in context of medical healthcare and what could be possible benefits? And with that being said, I would like to hand over to you, Professor Carr, and hope we all have a good evening.
Great. Thanks so much for the introduction and for having me. So let me see if I can share my slides. It looks promising and go into some sort of presentation mode. I think you should be seeing my slides now. Is that the case? Perfect.
Yes.
Excellent. So again, yeah, thanks for having me. I'm very excited. Also, thanks to those of you who took time to send questions. I already shared with Julia and Sebastian that the questions were super interesting. Actually, the questions are massively interesting. So I changed a few slides to address them, and I added a few studies that address some of your questions. Not all of them. So we'll go over that at the end of the talk. And yeah, my name is Andreas. I work here at the University of Florida. I've been here since 2007. I have a fairly international lab. There is always people from at least 5, 6, 7 different countries in my lab, which I really, really enjoy. And we study pretty much um, all different aspects that we can address with human systems neuroscience, including computational modeling and the tools of cognitive neuroscience to find, mostly using basic science, the building blocks that later we can then relate to mental health. Okay, so in the next 45 minutes or so, I want to go about some challenges. Mental health is really on people's minds. Since the pandemic, we saw a huge uptick in mental health problems. I think also the awareness of society that mental health is a thing and it's not a taboo, it should be addressed, should be talked about, is rising with every generation. Your generation is once again better about being active, talking and maintaining one's mental health. And I want to talk about the role of basic science in that framework. So I can't go into everything. I'll skip a lot of genetics research that other people do. My lab does do almost none of that. even though we contribute to some of that. So I'll talk a little bit about it, but not much. And I'm going to focus on things like neuroplasticity and computational modeling and the role that basic science studies can do in that context. We talk how we can use those basic science things that we have learned back in the clinic in a transdiagnostic way. I'm going to talk about what transdiagnostic means. I'm going to do a few conclusions, and then finally I go back to your questions, but I'll address them throughout this presentation. So hopefully you find something in there that is related to what you asked if you asked one of these questions. We have a lot of our materials for these overview talks on OSF under this link, and all our computer code is in this GitHub repository that's called CC Lab. That used to be our former center that my lab was part of, but that is no longer present. But we kept the name for just sentimental reasons because we missed the old center. Okay, so let's have a look. So if we consider mental health problems, one thing that we see and that's on people's minds, especially in the Western world, but also in places like China, where I see a lot of papers that I get for review where people say, look, there is a mental health crisis. So people use language like this, and you can see why that is. Overall, just under 25% of people in the United States, and this is fairly representative for Western countries, show any mental illness by definition of SAMHSA and NIH. And these are numbers from 2020 to 2021. Most likely they have increased a little bit. Mental health issues are overall more prevalent in females. If you use biological sex as a proxy, then they are in males. They are also more prevalent in younger people than in older people. And they're really, really prevalent when somebody has mixed race, mixed ancestry, mixed ethnicity. So if we look at, for instance, this combination, Native Americans, Alaskan Islanders, and another ethnicity or race here in the States, there is more mental health that's being reported. I know that other countries, especially Germany, has different ideas about ethnicity and race. It's different in every country. Here in the States, for a little while, there was an effort to increase representation and provide special services to people who are particularly disadvantaged because of their ethnicity and or race. And even though the current administration tries to get rid of that, I'm sure it will rebound because it's a very obvious problem that's all over our data. There is a few challenges with assessing and describing and knowing what the mental health problems are. So as scientists, we would really want to be able to say, oh, we have a firm grip. We know what mental health is. We know what mental health problems are. Here is our rules for assessing them, measuring them. And then once we find out what the given person has going on with them, we just do a thing and that will help them out. Just like your family physician does when you have appendicitis. They say, oh, you have appendicitis. I know what to do. We just cut that thing out and you're going to be just going to be right as rain. So we would want something like this, but we don't have it. And there is many, many different reasons why we don't have it. If we look at our diagnostic process, it is really as it is conceived of today in psychiatry and clinical psychology based on a conversation between two people. The two people being a healthcare provider and the patient. A healthcare provider may ask direct questions that are standardized in some clinical interview or may just go through the list of symptoms, and then the patient or client is going to be reporting on it. And then basically what happens is we count the symptoms that somebody checks off. So we have a checklist and we say, oh my God, this person has 6 out of 11 possible symptoms. And so that means maybe they cross a threshold or not. And then we say they have this problem. Then we give this problem a label and we say this is called social anxiety or major depression or something like this. So this is how this is going. Then there is some intervention. And the interventions include mostly in most countries where this is available, pharmaceutical interventions, that is maybe the mental health care provider prescribes a drug, especially when that person is a psychiatrist. In many cases, people will not see a psychiatrist. They might see a family physician or even a pharmacist, and then they'll get access to those drugs. There might be psychotherapy. There might be psychoeducation, which means we explain to the people what's going on with them and we give them some empowerment by knowing about their problems. We might introduce well-being activities to them. That's very common. There might be biofeedback, not as common, but a lot of places have those. Neurofeedback, also a lot of places do those. And then more recently, there is also brain stimulation and a bunch of more rare interventions. So how do we go from here to there? Again, if you have appendicitis, the diagnosis of appendicitis up here would imply what the therapy is. In mental health, many people would say, yeah, that is actually not really true. There is a little bit of a rule of thumb that experienced psychiatrist will say, oh, this person has OCD. I definitely want to give them a serotonin reuptake inhibitor, or this person has psychotic symptoms, so I'm going to give some sort of antipsychotic drug. But everything else is a little bit unclear. It's also a problem, and again, even in Western countries where people have access to services, and even more so in countries that are more poor, where people don't have access to services, where really only a minority of people even receive services. So there's a secondary problem, even in rich countries where people don't seek services or don't receive them, or don't receive targeted or expert services. There's another problem that I need to address also, and that is the heterogeneity of these report-based diagnoses. The poster child of this has become major depressive episode. You need for the DSM, this is like, for those who don't know, the DSM is really the Bible of making psychiatric diagnoses with clear rules, what the person has to say, and then what you count, and then when is the count going to be resulting in a diagnostic assessment where you say, yeah, this person meets diagnostic criteria. So I wrote that down here for major depressive episode. You need 5 or more of the following 9. Let's go through them. It's totally worth it. So you need depressed mood most of the day. You need diminished interest or pleasure, which we call anhedonia. You might need significant weight loss or weight gain. That's why it's red. These are the opposite, right? Most people would agree that weight loss and or weight gain is like 2 different things, opposite things. Same with insomnia or hypersomnia. Same with psychomotor agitation, where you can't sit still and you want to just run around, or psychomotor retardation, where you only want to lie in bed and sit around but not really move. Then we have fatigue or loss of energy. We have feelings of worthlessness and guilt. We have diminished ability to concentrate, so these attention type symptoms. And then we have thoughts of death, suicidal ideation, and or concrete suicide plans. So out of these 9, if you meet 5, you will get that label major depressive episode. That's going to be your diagnosis. Now, the problem with this is because there is 5 out of 9 and 3 out of the 9 have these opposite kind of things, we have 95,040 ways to meet those criteria and get a diagnosis of major depressive episode. Now, that is problematic in many different ways. You can easily imagine that now 2 people have the same diagnosis. There might be a person who has suicidal thoughts, but sits in the corner, doesn't really move, say they sleep all the time, and also has continuous weight gain. Then if they have a depressed mood most of the day, that totally gets them a diagnosis of major depressive episode. Okay, now there's this other person who runs around all the time, may experience significant weight loss, say they cannot sleep, is highly anhedonic, and probably has some sort of attention problem, And this person has no overlap really with the other person that I just described, but both of them will have the same diagnosis. So that is part of the issue. So a person might suffer and might explain to the mental health care provider what is going on, and they might just have totally different things going on with their lives, but they all have the same label of their diagnostic assessment. And that means that it's really difficult from that label to come up with a treatment plan that is targeted and that is specific to that particular person rather than to a huge group of people who can have one out of 95,040 different manifestations to meet those criteria. And there is also symptom overlap. So not only is it a little bit difficult to say a person has major depression, what does that mean? There is also not really a world where the majority of our patients have only one diagnosis. Most people, as you can see here, this is from the Comorbidity Project by McGrath et al. I have that here as a little QR code. Most people have more than one diagnosis once they are diagnosed carefully. So the median here is really people with two diagnoses. A lot of people have three, and still a whole lot of people have four plus. More people have four and more than have just one. So you could say having just one particular mental health diagnosis is really an anomaly by itself. It's more normal to meet criteria for at least two or maybe three of these diagnoses. So comorbidity, if we want to call it that even, is really the rule. It's not an exception. In textbooks of clinical psychology or psychiatry, people tend to have a disorder and it's described as a thing. But in reality, it's the rule. It's not an exception. If you look out into the world, people who struggle with their mental health, they have more than just one of these problems. Okay, so I could go on with problems with current diagnostic assessment and finding treatment targets and intervention, but let's leave it there and think about how do we improve that whole situation. So it would seem we would want to understand at a more profound level what is wrong with a person. What does that person prevent from pursuing a happy, healthy, and fulfilling life? And how can we assist that person in a more targeted way? Giving them those labels is fine and gives them access to resources in countries where there are those resources, but it's not really a clear, unambiguous pathway to receiving the best possible care, right? That makes sense. Do we have any questions until this point? Let's see, I minimized you guys. Let's see if I can maximize you also.
As far as I can see, there are no questions.
Okay, well,
No.
if there are any questions, there's a hand. Patrick has a question.
So, hi.
Hey.
So would you think that the high top ideas, like the idea of the high top model would be then a good result for the classification problem or?
Yeah, sure. So for those who are not aware what that is, some folks, a lot of clinical psychology researchers and some psychiatry researchers got together. Now it's already also 10 years ago and proposed the hierarchical taxonomy of psychiatry, where there is a hierarchical scheme where a lot of these disorders overlap, and it's definitely better and more accurate and it's empirical. It's definitely a step in the right direction. I would say it does still not explain everything, but it explains why disorders are overlapping and so similar and why there is so much comorbidity. And it's a great way of thinking about mental health, certainly better than the traditional way. I hope that answers the question briefly, Patrick. We could go into a really long discussion of high top. It's very interesting. I'm going to talk about another framework that's called RDoC in a little bit, and hopefully that will also address your point. But yeah, high top is something that I like to look at once a week, and it informs a lot of the research that we do. So yeah, it's very useful. There was another question, I believe, for a second.
Yes, I raised my hand. Um, I don't know if maybe you'll resolve the question, but what I just thought, coming from the field of clinical psychology and in my day-to-day work, obviously I'm working in Germany, and well, our thumb of rule when it comes to giving somebody a diagnosis has always been so viel wie nötig, so wenig wie möglich. Which would obviously inflate the amount of comorbidities, especially looking at my colleagues, which are psychiatrists. I can often tell that just by saying something, they've nearly got the ICD-10 code already written down in the system, just because we've been raised in a system where everything needs to have a name and everything has to have a diagnosis so we get paid. Would you
Right.
also consider that it's a systematic issue rather than I would say, like a systematic from a field of research?
Yeah, completely. So the reason why we have not abandoned those things and why psychiatrists in particular get very angry when you say, let's abandon things like ICD and DSM, which honestly very few people are saying, but they do get angry when you suggest that or when somebody suggests that because it protects the patients, like for a patient to get reimbursed, get access to services, and people get paid. This is the system we have, and it's not a bad system. I think it's mostly bad because it tries to do this practical job of people who work in the field as therapists, which I did for a year before I went back to academia and just decided I want to do more research. But I really loved for people to have what I thought is the correct diagnostic label. And then I loved for them to have access to services, which then normally arose from a discussion among the team of therapists. So that was kind of, how it went. So I agree with you. Everything that was implied in your question, I think, is true. And the one reason why I think we want to move beyond these diagnostic labels is we want to not have the research be based on them because we kind of know that they are not as sharp, not as accurate, and not as targeted to specific things that we can understand quantitatively. So it has held research back quite a bit comparing groups with depression with healthy people because the depression group is so heterogeneous is one reason. Sebastian, you had a question.
Yeah, I just thought about adaptive testing. Is that a thing that would be implemented in diagnostics? Because if we think about intelligence, for example, there's the ALE-3
Mhm.
that might improve it significantly, right? If you can readjust and not only count, okay, 1, 2, 3, 4, 5, may that be a thing? And also, is there research done on that topic?
Yeah, it's normally done under the umbrella of specific categories of mental health problems. So there is adaptive testing, for instance, in terms of startle response in PTSD, right? Like DSM says, if you have PTSD, you have a heightened startle response. So that's one thing that people said, oh, we can actually quantitatively and dimensionally and continuously assess how that is. So there is now people who do this in their treatment planning and in their treatment outcome monitoring, where they measure continuously and dimensionally people's startle response when they have PTSD. So there is the beginnings of it when it's really easy to do so, when it doesn't involve paying 1,200 euros, I guess, to put somebody in an MR scanner. People will do it. So there is like heart rate based things that people are doing. There is a little bit of EEG based things that people are doing where, yeah, they look into, is this changing? Is this adapting to whatever I do when I do the treatment? Or is it changing or adapting to anything the patient is doing on their own? And so that is used as part of the assessment array, but it's rare. Like a lot of healthcare providers don't have the knowledge capacity or the desire to use those more continuous measurements. Normally, they're more comfortable using questionnaires or just the unstructured patient report. And Lirim has another comment or question.
Yes, maybe an add-on from practical day-to-day work when it comes to assessing certain criteria. Obviously, as psychologists, we've been indoctrinated by questionnaires, like you just said, even though I'm more into like the neurological field of psychotherapy. And well, in the end, it boils down to I can read the manual for each assessment and dissect every single value and calculate specific t-tests for the single person I've got sitting in front of me. But then I would see one person a day and would write one assessment a day, which in a practical sense wouldn't make sense. That's why I'm so
looking
forward to what you might also talk about today. Maybe it will
increase
the speed of assessment.
This is the point where I have to think about how I best manage expectations here, because there is no simple solution to that problem. So let's just say you'll be in for a little bit of disappointment, and we will gather at the end maybe how much disappointment exactly it was. But yeah, it's tough. And when we do our studies with patients, normally we do this totally unrealistic patient screening first. That takes as long as another patient assessment, and then we do a 1 day complete assessment, which would never be realistic in any paid or non-research context. But we do this so that we can see what of our array of neurological, if you so will, and peripheral, autonomic, and other quantitative measures map onto this really careful assessment. But yeah, whenever somebody sees our assessment, they say, yeah, you spent like 8 hours with the patient to assess them. You are crazy. We could never do this outside a research context. And it's entirely true. So yeah, thanks for that. That was a really important contribution. So let me go back to the beginning and switch gears. So from this very cheesy sunset picture, this is an old building here at the University of Florida that's very ugly. This picture is also taken in 2001. We moved out of that building last year because it had to be abandoned for being entirely molded in the inside. This is in a swamp. And in that building, people come up with a really good idea that led to this research domain criteria project. And Bruce Cuthbert was the NIMH director at the time. That's the National Institutes of Mental Health. And my former colleagues, both now retired, Peter Lang, Margaret Bradley, really formed the foundation of what we now call the Research Domain Criteria Project. So here is what it is. It's just the idea that for research purposes, for a moment, we want to assume that we can do at this point some research that starts linking the environment of a person with their own development and looks at different levels and dimensions in the body, in the behavior, and examining the human's mind with the measures that we have available to us at the level of genes, at the level of molecules, at the level of cells, circuits, at the level of physiology, at the level of behavior, and at the level of self-report, and try to look at this in particular, what the RDoC system calls domains, which are really areas of behavior and experience that we can understand in a comprehensive fashion. So negative valence is everything about my behavior that tells me that something is bad, threatening, anxiety related, or something like this. Cognitive systems, you guys know, would be things like attention, working memory, systems for social processes, arousal systems, like there is those systems. We kind of know what they are, but now we want to understand them continuously and dimensionally. Ideally, we want to get an idea, at least hypothetically or in a theoretical framework, how they are arising over time as a human being goes through their lives at those different levels of observation that are here in this green bubble. And we want to kind of describe them as much as we can. One key in this is neuroplasticity, because we kind of assume how that will change over time as a person makes certain experiences. So the very ambitious idea, and people like Tom Insel and Bruce Cuthbert and now Sarah Morris, who's in charge of that program now, they all know that this is very ambitious, but the ideas are relatively straightforward and most people can come on board with those. So we want to use basic science tool to identify more dimensional measures So I don't want to say Sebastian here has PTSD, which hopefully he doesn't have. I want to say he's easy to startle, and he has nightmares, and he has a very, very low threshold for being defensively engaged. None of which is true, but I was tempted to use an example. So now I have described him on 3 dimensions instead of just saying he has PTSD. Another person also might have PTSD, but they might largely have a substance problem that I can measure. They might have even more heightened startle response, and they might have a low threshold for delusions and maybe some idea for psychosis, which might be related to a slightly more susceptible dopamine system, which their dopamine system could be assessed using nowadays even magnetic resonance spectroscopy or something like this. So I could come up with dimensions that I can measure, and now I can describe each person that sits in front of me if I have enough money to put them through all these dimensional measurements. Again, today, practically, we don't have that. We could not say, let's do that with all the patients. Nobody would know how the money comes from, but we also wouldn't know exactly what to do. So this is all for research. We would focus on mechanisms and finding new treatment targets. So if we find out that somebody, for instance, has a tendency to respond to even imagining a certain stressful situation with a heart rate acceleration, whereas others respond with a heart rate deceleration, where the heart rate slows and they freeze, we would say, oh, these people do different physiological things in response to what they both describe as threatening. But one person's heart rate goes up through the roof and they almost like freak out. The other person's heart rate drops and maybe they almost faint. So they do totally different things physiologically. Even the self-report might be very similar. So we have now a treatment target that we didn't have before. Before we just had what they said. Now we know there is like totally opposite things they do at the level of the heart rate response when I tell them to imagine whatever bad thing it is that they are having difficulty dealing with. The RDoC also emphasizes neuroplasticity. I have that here as this axis. And the idea is to come up with more refined assessment and more tailored interventions, and that these interventions may include physiological intervention, even neurophysiological intervention. And there is a few examples where this has worked, or even existing neurophysiological interventions like TMS, which is transcranial magnetic stimulation processes have been understood better as a consequence of RDoC-based research. You can apply RDoC to a lot of stuff. Again, this is just another example of how you can have a patient with social phobia and how likely it is that they have all these other diagnostic categories, the criteria of which they also meet. So you have a bunch of people with social phobia. Very likely they have 1 or 2 of these. And now, based on those, you could come up with dimensions that you want to look at instead of just listing all these different things. This is a paper that just came out earlier this month, early 26, came out in Nature. This is Kenneth Kendler and his people. First author is Grotzinger. And they studied a lot of genetic material from a lot of patients. I think it was a few tens of thousands of patients. And they came up with these clusters of symptoms, compulsive disorders. I forgot what SB is, if somebody wants to look it up. This is substance use disorder, internalizing disorder. This is neurodevelopmental disorders like ADHD. Oh, SB is schizophrenia and bipolar. Yeah. That always is its own thing. They're very similar. Don't spend too much time looking at this. They did a high-top-like approach with this. The people who asked about high-top will recognize the overall idea that there is a general factor of not being too well in terms of mental health, and then there is one level. Is there a question?
No, somebody forgot to mute herself.
Oh,
So
yeah, that happens.
I just did it.
That's perfect. No worries. I just don't want to talk over a person with a question. but people can have their coffee break in the background. That is completely fine by me. Okay, so there is these 5 different groups of disorders, and they break down into more specific groups, again, of diagnostic categories, which, again, often they group that way. This is how they group in the HiTOP that was mentioned earlier, the hierarchical taxonomy of psychiatric disorders. Why I put this plot here is because of the purple stuff. You can see those pie charts. In each pie chart for each disorder, there's purple stuff, and that's the variance that is not explained in the genetic variation by the particular disorder at all. So this is shared variance. What was the major finding, and you can kind of see that here in these correlations between those different higher level groups of disorders, is that they are still super similar. There is a 0.42 correlation here. There is a 0.2 correlation. There is a 0.55. There is a 0.6 correlation. Also, the correlations across a few, they're all very high, 0.74, 0.47, 0.44. All of these 5 major groups that are supposed to be independent, they're still highly similar in terms of their genetics. So what we're seeing if we do genome-wide association studies in that field is that even at the level of genetics, when we have the study set up to detect the specific problems that a person has, what will be found in many cases, and this is the largest study that has been done so far, what will be found is that all these people are very similar. So it's not like, oh, there is this very specific genetic risk profile that maps onto bipolar. And it's totally different from the other risk profiles. That is not what is being found. What is being found is that a lot of people have a whole bunch of different risk factors genetically, and they may manifest in many different ways in terms of their actual diagnosis, how it presents itself when these people talk to a person who makes a diagnostic assessment on them. So this is very important. It's just really great data. I have the reference here. It's also beautifully written paper. So what my lab has decided is that we need to tackle this in one particular specific point, and one point that I really like is fear and defense. It's at the core of a lot of mental health problems. It can take the form of exaggerated fear responses when a thing is actually in front of you, but it also can take the form of worry, apprehension, avoidance, where I don't go out of the house because I'm kind of afraid that something might happen. Right off the bat, that has a lot to do with the questions that some of you asked, where a fear and defense response is adaptive in nature. Like there were a few questions like this. Obviously, I live in Florida. If I don't have a fear of alligators, I would not be sitting here doing this talk because I love kayaking. This is my spouse, Stephanie. We're in a beautiful mangrove swamp. They are paddling right through. And you see all these branches. I would say it's fair to say that on every 10th of these branches, there's one of these little guys. They're called the Florida cottonmouth snake. They're very poisonous. They have the same poison or venom, I should say, as a cobra. So you don't want to get bitten by them when you're far away from a hospital. So you get used to that, but you also don't go near them. And this is an adaptive fear response, right? You don't want to be fearless when these guys are around. When you see one, you want to make a defensive move, which consists of leaving enough space. So if they're on the left, you pedal right? That's your adaptive fear response. It's very clear what that is. And people have studied that a lot. There's a lot of animal model research in all senses, not just vision. And then people, based on the rodent work in the field, which is very strong, they came up with this standard model of fear and anxiety. The standard model is such that you see a thing, and then eventually that information, either fast or slowly, it gets to the amygdala. The amygdala has many efferents to all over the body, and it creates a state of hypervigilance. It heightens defensive reflexes like the eye closure or the kind of fear and loud noise reflex. It also strengthens the patellar reflex. All defensive reflexes become amplified. It releases or facilitates the release, I should say, of stress hormones. It prompts fearful thoughts, feelings, and behaviors. It heightens cardiac responses. It prompts electrodermal responses, and it also prompts defensive motor action as appropriate. Now, if you think about this as you can already, from your own experience, see that if I show you a fear cue, your actual defensive motor action will be very, very specific to the fear cue. And actually, it might then not come as a surprise that all these other physiological responses also depend on the meaning that that fear cue has for you. And this often can be very striking. So if you're a person who doesn't like needles and is a blood and injection phobic person, and I show you like now very surprisingly this big picture where somebody is penetrated by a needle and maybe there is blood, then your heart rate actually will drop. Your blood pressure will also drop. And you might also be close to a world where you faint because of this. So your cardiac fear response will be one of heart rate deceleration, lowering the blood pressure and keeping the blood in the body because you're a blood and injection phobia person. If you are a dog phobia person, and I show you this picture now, or a movie where a dog bites somebody in the leg, your heart rate will go up and your blood pressure will rise as a consequence. So it's the opposite again, but both of them are fear responses to a very specific group of stimuli. So even in this world, these things are not fear responses that can be labeled as a fear response. They are fear responses to something. And because they are defined by the something, they might actually not be the same. So when your amygdala says this is a fear stimulus, that doesn't mean that your heart rate will go up. It means that now there is a defensive response in your physiology, in your cognition, in your emotions, in your behavior that is adaptive for addressing that particular thing, whatever that is. And it depends on what you have in terms of experience with that thing, how that defensive response will look like. So it's not hardwired in that sense, right? Because it really depends on what that stimulus is. It's also not so simple because, again, it varies with culture, it varies with your expectation, and it varies with the type of fear and what you're fearful of. So to me, this is very interesting. Environment and culture really rule heavily in this. And this is not just some vague, saying, oh, everything is depending on culture. You can show that your heart rate will drop if I show you the needle and you're a blood and injection phobia person. So it is a very hard physiological truth that fear is not a universal response that is dumb. It's a very intelligent, very flexible, very, very shapeable and malleable response. Okay, I think I made my point. Now, a lot of what we know about this comes from rodents and also marmoset monkeys. So these are the stars of the standard model that I just described. But in humans, unfortunately, it becomes even more complicated because in humans, the amygdala alone is not necessary for human fear and defense. There's people with no amygdala whatsoever. They show really strong fear and defensive responses. There is also very limited evidence that if you do some imaging, even invasive imaging in people with implanted electrodes, that their amygdala is specifically responsible for any fear or defensive behavior. In fact, one of the safer way in humans, even in an fMRI study, to get the amygdala to get activated is to show a face or erotic scenes or anything that's highly pleasant, and the amygdala in humans and primates will get engaged by this. So we can't really rely on the standard model where the amygdala alone is a hub for defense and fear and anxiety, because in humans that is not true. The amygdala in humans is engaged in a lot of appetitive, pleasant processing, and it's also not a necessary condition for functioning at the level of defense and fear. So in effect, we learned a lot from rodent work about fear and anxiety, like Joe Ledoux is a big author in that field, who also did his work in auditory systems, which are slightly different. Some of you had questions about that, and those are great questions. So a lot of what we know from rodents, because rodents don't really see or use vision as much as we do, we know a lot of that from hearing. And in humans, we have to build on those things for both reasons. A lot of the work is not visual. Humans are a little bit more visual. They're also auditory. But these other reasons that I gave suggest that for understanding fear and anxiety in humans, we have to kind of go beyond the standard model that's purely amygdala based. So do we have questions until that point?
So I might have one, but I guess I wait like 30 seconds so one can process all you've said until now. But yeah, maybe I just fill in the gap. So it is also referring to a question that I've written and sent to you before. It has been submitted. But you've shown, or you could show that the system behind all the neuroplasticity changes on the plasticity, but the reaction in EEG, for example, changes during fear conditioning. If we look at certain things like amygdala and if you model these systems, then have you ever compared like the pre-post model of a person that is fear conditioned, isn't, and then also a person with a strong anxiety disorder? And is this maybe a way that we could use such modeling techniques actively in psychiatric diagnostics?
Yeah, I think it's too cumbersome to use as it is today. But yeah, that kind of work has been done by me and many other labs. And it's very promising, I think, because fear conditioning is such a fundamental process. Now the problem becomes, and people are working on this, how can you have a fear conditioning paradigm and the associated measurements in a way that is compatible with running a lot of people in a short amount of time and learn about things like neuroplasticity. Learning about neuroplasticity and short amount of time in the same sentence is a little bit of a problem because the neuroplasticity takes a lot of time to take. I show that to you in the next few slides, which will go a little bit faster, where we go over data. But yeah, that's a great question. I think I have some more data that will speak to that probably.
But may I add just a little something?
Sure.
My question wasn't all about the plasticity of a process, but I assume if there are some similarities in the EEG pattern compared between those people with anxiety disorders and post-fear conditioning, then you could also apply these ones as criteria and use EEG more actively, but can be done maybe in just around about 2 hours, would at least be more applicable than targeting the plasticity as a point, and then look at the pattern generally in people where you might think they have an anxiety disorder to prove that.
Yeah, no, this is a great point. So I think the short answer, and I don't know if I have a slide on that, but if I were to do fear conditioning on you, for instance, and it would succeed, and I had enough times, enough trials, then after a little bit, you get, tuned to the CS plus. The CS plus is the condition threat queue. And there is different ways I can measure that tuning. After doing this, it would look like a patient with a specific phobia, for instance, that hasn't had the specific phobia for a long time. And I say that specifically because as chronicity and severity interact over time in a person, these biases start looking way different. And I have a little bit of a slide about this later. So if you had it for a short amount of time, maybe high severity, but low chronicity, also low comorbidity, PTSD, specific phobia, social phobia, something like this, you look like a person who just has been fear conditioned. The longer you have that mental health disorder, and yeah, we have that now from hundreds of patients from Florida, the more you start looking different again, because the fact that you have this disorder changes you over time again, and it changes along with changing your behavior, your approach to your problems, it also tends to change your markers of plasticity and your physiology. So that is very interesting. That's part of our job. I see Lirim has his hand up.
Yes, I wanted to elaborate also, that I think throughout the disorders, there have been a lot of studies trying to prove exactly this. I can remember a study like in the beginnings where this whole topic of emotion induction was happening. People tried to induce negative emotion and then prove that it looks the same as in a depressed person, and it doesn't work. It looks like it for, like you just said, it looks like it for a little second, and then all of a sudden you see developments which you can't explain via this paradigm you use. And it's, I think, something that's been holding us up for, at least the way I know it, for like 30, 40 years, ever since this emotion induction thing has been going on. And we still don't really know why this is happening.
Yeah, no, that's exactly right. And so hopefully we can have some conjectures later about why that is, at least in one particular domain. But this is a really good description of this. All right, then I'll just go there. So now we can go through data. So we can measure fear at many different levels. We can use the brain, the body, the environment. We can measure all these things. And we can look at visual cortex. We can look at population level postsynaptic signals. That is like human EEG. There is also some intracranial EEG work. Increasingly, these data are available to all of us because they're shared by large consortiums. And let's see. So I like to start with first contact. Again, I live in Florida. This is one of my favorite examples. I grew up in southern Germany at the border to Austria and Switzerland. There were not many alligators there. But here in Florida, you have this problem, I should say, which is when you see a thing like this, it becomes like a little bit of conundrum. Is this an alligator or not? If it's not an alligator, you can step on it. So this is very nice. When you're in a swamp, you want to step on logs and keep walking. I love hiking. I like being out in the prairie and in the swamp. But if it's an alligator, you should not step on it. That is self-explanatory. Why not? So the question is that one or not? At the beginning, when you move there from Germany, you often cannot tell. Here, the giveaway for those who must know is that little tooth here. Most logs here in Florida don't have teeth, but this one has a tooth that stands out in the right way. So definitely we've established this is an alligator. There it is. It's exactly that alligator, of course, and we've established that. So if you live here for a while, you establish and develop these visual skills to scan the environment for things like snakes and alligators that don't exist maybe in southern Germany as much. So that's actually represented in the research. The people who wanted to study fear and anxiety often have these papers, and I kind of collect them. This is just a small, small selection of them, where they said, oh, we wanted to study emotion, and we were looking for amygdala stuff. And by the way, the biggest difference between whatever we studied, it was always in visual cortex. It's kind of hilarious reading those discussion sections of those papers where they said this is from the Boston fear people. They had a very large treatment outcome study, and then they found visual and attention regions were the ones that predicted treatment outcome. This is more recently. There's a bunch. I mean, you can find them where people write these things and they say we had these high hopes and maybe it was in prefrontal cortex and fear must involve the whole brain. But then they say, yeah, we found it here and there, but mostly we found it in visual cortex. It's kind of really funny to me. So let's go back to these people and look at how we frame this. So we look at the visual system not as something that's particularly relevant, but because it's also evolved to support survival. It's just part of the human defensive repertoire. It's not just like a camera that takes a picture and tells the rest of the brain to interpret it and do something with it. If it's a fearful thing, it's part of the defensive repertoire that we have. That also means that it has to adapt. So it needs to change, just like when I moved to Florida and there were alligators, and now I scan the environment effortlessly for alligators and snakes. It will adapt to whatever environment a human being finds themselves in. That means it's shaped by experience, and it also means it retains information, so there is a memory component in it. These are all very, very, clear, but people just don't think about vision in those terms normally. So it took me a lot of convincing at vision conferences and stuff for people to become interested in that. One thing that I'll talk about today is visual biases. So if I can maybe put a bias into your visual system that would allow me to examine in a laboratory environment those things. And basically, that's what I'm going to be talking about a little bit. We're going to be looking at different timescales, dozens of trials, hundreds of trials, thousands of trials, and days later. So we have data on all of those, and that kind of helps us understand how things change. The idea is that there is an initial acquisition of a bias where the whole brain communicates and tries to figure out what that new thing is that is coupled with threat. Then there is sustained learning when it comes up again and again, and I realize that this is now this is part of my life. And then maybe after days, weeks, months, years, there is a persistent threat bias, just like with the alligators. And that is mostly residing in visual cortex. So you can see this is your visual cortex. At the beginning, it requires a lot of communication across the brain. And then there is some attention process later during sustained learning, and more the visual cortex becomes important. And there is then in the end the ability of some neurons in the visual brain to respond to those without much back and forth with the rest of the brain, because they figured out that these features, even though they might be low-level features, are always coupled with bad outcomes. So this is basically just learning and plasticity at this point, and it takes time. Okay. So there is mathematical formalizations for all these things. I will not bother you with those, but if you're interested, these are all very clear, very simple, all well established. The only new thing here is how we combine them. Perfect. So let's skip those, get a little bit ahead. And to study vision, we flicker a thing that drives what's called a steady state visual evoked potential. And don't ask me about those, ask Sebastian about those. He knows all about them. There's another thing that's called alpha power reduction. You can measure that with EEG on the scalp. And it's very interesting. We found in my lab that it's extinction resistant. So if you try to extinguish the fear through extinction learning, the alpha power reduction stays for a long time. I'll show that to you later. And we also do this simultaneously with fMRI to see where the sources in the brain really are, where these visual and more attention-driven changes are. You can see alpha power reduction is more smeared across the whole back of your brain. It includes visual, but also parietal regions. The steady state visual evoked potential, a flicker response, mostly in the way we use it, or pattern reversal response, is really a primary visual response, really much just focal at the back of the hand. Okay, and now we need a paradigm, and the paradigm is so simple. It's painful. We can do a flickering of a CS plus, and here this is just a patch-oriented grading, oriented in one direction. And then one of them always leads to a bad outcome, like an electric shock or loud noise. That was on the last slide. And one of them is always safe. So no US. So people just learn this is bad, this is good. We do it all over again. It's very simple. Sometimes to confuse them and to measure like tuning, we throw in generalization stimuli that are like neither of the two, but they vary in similarity with the CS plus. Does that make sense? Okay, it's very easy. Okay, let's look what happens when we have dozens of trials. We measure the SSVP. After like 10 trials latest, most people show a heightened visual response to the CS plus. So this is before learning, CS minus, CS plus, those two gradings, they're both boring. People say, what the heck, it's just light, And then after learning, the CS plus becomes more important to them. Now it's also increased in visual cortex. You could call that attention. You could call that heightened perception. I'm actually not quite sure what's the best way of talking about it, but it's visual cortical facilitation. Same with alpha power reduction, preconditioning, post-conditioning. The alpha is an ongoing oscillation in your visual brain that just goes on. when something relevant happens, tone, picture, it gets blocked, so it gets reduced in power, and it gets reduced more when that thing that blocks them is a CS plus as opposed to a CS minus. These are different versions of them. All the dark ones are CS plus. So you can see post-conditioning that also is increased. And when we look where in the brain that is, we can see it's actually quite in many different places of the brain. So there is a lot going on, and it also is related to the heart rate response in response to the CS plus. A lot of people show an initial deceleration where the heart skips a beat, slowed down, then it accelerates. So this is your ratings. If you do this generalization thing, this is the CS plus here in the middle. People kind of learn that, and then they say, yeah, I don't like this one when you ask them after conditioning, and the others kind of gradually go and are liked better. This is displeasure. So after conditioning, even when they're all very similar, you can see these are quite similar. They're only 10 degrees different from the left to the right. This 45-degree one was the one that was paired with an electric shock. So they don't like that one. That makes sense. Let's look at their visual cortex. Before conditioning, the visual cortex says, I see a lot of these things and I don't care what they are. So everything is the same. There is always a response in visual cortex. After conditioning, the CS plus, which is this one here in the middle, it becomes amplified. The most similar ones become inhibited, and the other ones are pretty much left alone. So this is a pattern we've seen a few times in many studies, and that happens really, really early on in the trial. We call that sharpening or Mexican head tuning. There's different ways you can address this. It's something that's not a huge effect, but it's there normally when you have enough people. It is related to information coming from the front, from frontal sides. You can look at phase locking, for instance. I'm not going to go into any really detail here. So after dozens of trials, and this is just a trial by trial representation of this, what we see is that there is an amplification of the CS plus. It's shown in red. This is the CS plus here. These are the similar simile. It amplifies, 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 and then similar cues are being reduced. In extinction, that whole thing turns around into its opposite. We found that particularly super interesting. I was really not ready for this when we did this the first time. Where now the CS plus becomes inhibited and these become disinhibited. But you can see here that Mexican head tuning where the similar ones become decreased, and that actually the increase gets larger and larger as learning progresses. This is trials here on the x-axis. So we see sharpened tuning in visual cortex, basically. Some of you asked, how about auditory cortex? It's very similar in auditory cortex. There is even better animal data in rats where their auditory cortex works exactly like this. Lirim, by all means.
I was just, I don't know, you just said you were a bit surprised when you had the extinction pattern show up from a psychotherapist's view. When I'm thinking about, let's say, specific phobias, I'm doing a stepwise exposure therapy. The funny part is this procedure is basically what we do in stepwise exposure. We start with the main stimulus, and then we've always been told to make it work towards the future, we have to use these like common stimuli and also expose the patient towards them, which I think is funny to see in a brain scan, basically in bare bones science, what we've always been taught how to do in practical sense. So to me, it seemed very obvious that would happen. It's I find it funny to a certain degree.
Yeah, I know. It was also funny to us because when we had this finding, this was still with Peter Lang, who since 1958 came up, together with Joe Walpe. They did these studies on desensitization and fear hierarchies and systematic fear exposure in that way. And they always had hoped to see something like this in any physiological data because that would mirror the way the instructions go. And the treatment target idea that underlies systematic desensitization. But in heart rate data, pupil data, skin conductance data, that is normally not seen. So I didn't really have it on my bingo card that it would be seen in the visual cortex of all places. But I'm with you. Yeah, of course, this mirrors like how people go about framing their systematic desensitization, where they say, yeah, you want to change generalization patterns in that way. It's interesting. But yeah, it was entirely unexpected. Just because we have from the same experiment, we have skin conductance and heart rate and startle, and they don't show this at all. So that's what's been the issue. But yeah, great point, Liram. Okay, so what happens after hundreds of trials? And now we look at alpha reduction, a metric of what we thought is attention. For hundreds of trials, we just now have a line that shows the alpha reduction. This is a CS minus, this is a total safety cue, and alpha just goes up. Alpha is really when your brain is bored. This is a really painful shorthand, so a lot of people would get angry at me if I let it stand like this. But just imagine it, there is more alpha when your brain is bored. This is not entirely wrong. And now let's see what happens with the CS plus. So in habituation, before we do any conditioning, it's just the same. And then the CS plus just keeps going down, 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 down. And it keeps going. Like with the learning, it doesn't really just do this at the beginning where you pay attention to the CS plus or you pay attention to the contingencies. It keeps going more and more down as much as you continue the conditioning regimen. And this is a generalization stimulus that's neither the CS minus and the CS plus. It's in the middle. It's similar to both equally. And it actually ends up in the middle, which is nice. So we can understand this. Now, this is when they become aware of the contingencies after, like, in this case, just 12 trials. After 12 trials, they say, yeah, sure. I understand this now. This is the CS plus. Whenever I see that, I get shocked. This is the CS minus. Whenever I see that, I don't get shocked. And these are similar ones. So here it happens. But really, after they find that out, their brain keeps learning. It's not like they find this out and then this is it. Whatever they say about their state of awareness doesn't really have anything to do with setting the level at which the brain operates. It continues to change after that quite a bit. If you wonder what that bump is, this is when we go from 100% reinforcement to 50% reinforcement, where only every other CS plus is paired with a loud noise or an electric shock. So yeah, so this was very interesting, also unexpected to us. There is just so much evidence for ongoing learning, even though at the level of awareness, they figured it out after 12 trials. And then if you say, no, but we keep going for another 120 trials, they will still learn at the level of the brain. Their physiology doesn't show any additional learning, but their brain does. Okay, so we saw that. There is ongoing reduction. I didn't show you some of these things, and I'm also slowly getting concerned about time. So there is an early response where the entire brain responds, and the longer I train, the more the visual cortex becomes involved, including attention and primary visual areas. One mechanism that underlies that we've studied in animal data and in human data is sparsification, where if you look how many voxels in your V1 contribute to the separation of CS plus and CS minus in some discriminant analysis, the number of voxels becomes fewer and fewer as the learning decreases. So that information becomes more and more confined into smaller areas of the visual cortex. They kind of learn this, but other areas don't learn it as much. So this was a fun finding that we did with my colleague Mingzhu Ding. So the number of diagnostic voxels decreases as learning progresses, even though the amplitude increases. And then this is probably the last I'm going to be able to do, and I can skip over a lot in this talk, but what happens after thousands of trials? So I had this grad student, Nina, did this study where she kept people in the lab for many, many hours in one session and conditioned them over thousands of trials. And what we did was we looked at the very early, very finicky potential. It's called the C1. It's an ERP in EEG. It's retinotopic, so if you see something below fixation and above fixation, it has opposite polarity because it engages either the lower bank or the upper bank of your visual cortex, because that part of the brain is retinotopic, which means it reflects where things are in the world relative to your fixation. So it's easily recognized. It happens after 70 to 90 milliseconds, and it is an efferent potential. It's just the first response in V1, in the primary visual cortex. We wanted to see if after many, many trials that changes too. So we did this kind of thing where there is only one place in the brain where neurons are eye specific, location specific, and orientation specific. And that is not in the thalamus because thalamus is not orientation specific. It's only in V1. So if you find any conditioning effects that are orientation specific, but also location specific and eye specific, that effect has to be in simple cells in V1. It's kind of funny because they're the only cells that have those three properties. So we said, oh, we're going to be figuring out if the learning happens in the thalamus, if it's not orientation, but eye and location specific, and so on and so on. And we did this whole paradigm where we did many, many trials. It was a 3.5-hour conditioning session, very, very not pleasant for the people. But somehow Nina kept them in the experiment. So this is the baseline. There's 24 people here. You can see that amplitude reversal. This is the right eye, this is the left eye. This is the upper visual field, the lower visual field. In this subgroup here, this is going to be the CS plus here on the bottom. It's a combination of right tilt, lower visual field, and left eye viewing it. And so only that combination was ever, but always, paired with a loud noise that was very aversive. If you wonder about the loud noise is worse for people than electric shock. People really don't want loud noises in their life. And now let's see what happens after thousands of trials of conditioning. This is 1,200 trials later. And you can see there is like a small base factor is like 6.8. The p-value in just traditional analysis 0.02, but there is evidence that, yeah, you keep doing this over thousands of trials, that early response becomes larger. And we replicated this with other configurations and in other people, and in general, we were able to find this. Also on day 2, more change. Like if you bring them in days later, the next day or several days later, there is still the alpha reduction and there is more change in the visual cortex. So just to make this not so specific, there's so much evidence that the change just keeps happening. If you keep conditioning people, if you measure them later, you bring them back, there's all manner of additional changes in the brain. It never ends and it never goes back to baseline. As Lirim said, when you do additional learning, you just heap on additional learning. You never go back to the baseline. And so this is the model predictions. We have the computational model. It just shows what I showed you earlier. And I should probably, yeah, I'm going to skip the deep aversive conditioning and I go to maybe this. Do I still have 5 minutes or do you want me to shut up?
Oh, no, you can definitely take your time. I mean, it is only the general thumb rule that we say 1.5 hours. So as long as you would be fine with, you could go on and explain also the deep aversive learning. So we are not like cutting you off as if you overdo it by 1 minute.
I really like the discussion. So normally I time my talks, but I like discussion much better. So yeah, I'm going to go through those and then do the questions. So I think I will be done in 5 more minutes. It's really easy from here. But I'm going to skip the deep architecture. It's not so interesting. We have this work with deep computational architectures that also show these similar effects. But let's go to the clinic. That's much more straightforward. So if you compare people who are students, say they're a little bit more socially anxious than others, but otherwise are doing fine. Everything in their visual cortex makes sense in the light of what I just said. It's kind of beautiful. So you take these still okay students, you show them like fearful faces or threatening faces or even gabors or something. This is their social anxiety score. And the people who say they're more anxious, they respond more. These are all the red dots. The green dots are the people who feel safer. They just overall respond less. It's not a gigantic correlation, but yeah, we found it in many studies. It's like just a little bit of a correlation. You can flicker a face for the social anxious people. You can do a Gabor patch with the GAD, that's generalized anxiety disorder people. It's just this tendency. So we thought, wow, this is the first time that something makes easily sense. The people who report more anxiety about these things, their visual response is larger in response to those things. Very easy to remember. But it stops right there, the making sense part, of course, because this is real data. So why would it be easy for us? And it started when we looked at treatment-seeking people that varied in chronicity. So here, this is treatment-seeking people from the clinic. There's two types of social phobia, more specific performance anxiety, people who can't give a talk really. And then there's these generalized social phobia folks. This is not the DSM distinction. This is a distinction that a lot of clinicians here in the US make, because the generalized people, they don't want anything to do with people in general. They are just overall very anxious around others. Now, if we show them happy, neutral, fearful, angry faces, the specific social phobia people, they respond more to the fear and angry faces, but not the generalized one. Which is funny because the generalized ones are really the ones we're worried about. The people with a little bit of performance anxiety, they're fine otherwise. They just really don't sign up for like giving the first presentation in the semester or something. But really the generalized ones have much more severity, much more comorbidity, and much more chronicity also. They're also older. And you can see they don't make a difference here. Like for them, happy, neutral, fearful, angry faces, everything is the same. If we compare fearful versus angry across the head, you can see the circumscribed performance people, they show a heightened response for fearful and angry, but really not these other groups. So then we stratified them and looked why this is happening. In the not impaired people, if we don't care about the actual diagnosis, control, panic, circumscribed, generalized social anxiety, these are the colors in which we have them. Then we see that increase that we saw earlier in the students, right? So not impaired does not show any amplification of angry faces. Minimally, their visual cortex also doesn't do much. If we go in moderate world, now we have a few generalized people, we have a few circumscribed people. We also have a lot of panic and agoraphobia people. That was our clinical control group, and they are heightened. And the markedly people contain a lot of generalized social and also some circumscribed social people. So this looks like the students I showed you earlier, where just the more they report about their fear, social anxiety symptoms, the more their visual cortical bias towards threatening and fearful faces is expressed. But now what happens when we look at the really group within these patients that we're most concerned about, which is the severely impaired people, they actually do the opposite. So there is not a linear thing that researchers would want where they say the more you have of this, the more you have of the other. It goes on linearly into this world where there is marked symptoms. But when we go into the territory of severe symptoms, then the effect reverses and we have a bias actually now towards safe things and a bias away from threatening things. And that's something we replicated in a much larger sample. This is now way more than 300 patients that I collected with the chair of psychiatry here with a large NIH grant, where we also modeled the data with a simple mathematical model that's shown here on the left. And again, you can see with severity and chronicity, you get first an increase of the visual cortical response. But then as severity and chronicity become larger, people have more problems, more comorbidity, more chronicity. The effect reverses, you see more variability, but it goes in the other direction. And that is something that we keep exploring and that we're really interested in. So let me summarize this. So across studies and measurement modalities, we saw that there is a lot of retuning and adaptation in your visual and attention system, and it emerges as you learn and keeps actually emerging, keeps changing. Prolonged learning just makes additional changes. The emotion structures, and I didn't talk about this as much, as the amygdala become less and less obvious in the data. Insula is very strong at the beginning. Amygdala is very strong at the very beginning of conditioning, and then kind of people don't find it. My lab was part of a very large mega-analysis of 1,800 people's fMRI during fear conditioning, and there was no amygdala. So in humans, amygdala and fear conditioning are not very strongly related. But what is related is the emergence of these more sparse and effective visual networks, just because humans are very strong visual animals. Again, this is also true for other things, auditory, somatosensory, there's just much less work. And when we use computational models, we have a few of them, then we can compress all the complicated data that I show you into few parameters of the model. And those model parameters could be, they are interpretable. So they could in the future, and I would say in the quite far future, could be treatment targets and or could be used for intervention monitoring. At least they could give us an idea how to talk about these things in a quantitative and dimensional manner. Okay, any questions about this? Because then I go into your questions afterwards. Let me see if I can see you guys. I make this larger. Go back.
So just tell me when you're about, because I have at least one or two questions, but
Sure, let's go back to this and then let's do questions.
yeah, I mean, as last time, I may fill the gap in until the other people have processed. So the model basically that I've shown for the adaptation of the audiovisual system, isn't that basically this is just a smaller question, the sharpening model that was introduced like in 1989 by Martin and Weeks? Because this looks so similar to me with these dots that decrease but increase in power, and then so you reduce the activity, but while you have learned.
Yeah, exactly. So there's many such sharpening models. It's not at all new. The only thing that we have added to that is that we say where would it happen in the brain and based on what feedback and when would it happen through what interaction and what is the mechanism that that makes that happen. So we also predict that it would only be in V1, for instance, because V1 is the only place where you have these lateral inhibitory interneurons that would make that kind of Mexican head pattern. It wouldn't happen in other brain regions where you don't have that setup. It would not happen in hearing, for instance, which is true. It doesn't happen in hearing. You get retuning in hearing, but it follows the way the auditory cortex is set up, which is not through lateral inhibition of neighboring frequencies. So you need lateral inhibition between neighboring things on your feature. If you use a feature that isn't orientation, but for instance, spatial frequency, we also don't get any Mexican head tuning. So we just enriched existing models and made them more mathematical and injected some neurophysiology. We didn't have to invent anything.
Yeah, I just had to ask. There was a model that was mentioned by
totally.
Professor Gruber in one of his lectures, so therefore.
Yeah.
And
No.
he primed me before we started today, so.
That's very good. No, there's also like the feature gain similarity model by Martinez Trujillo and Stefan Treu, German person. That's also very similar for attention. So, like there is a lot of stuff for us to draw on. There is a bunch of models that we combined to describe our data, for sure. Absolutely. It's really nice. Like you can build on so much if you do science that's already there. If you know the literature, it's shocking.
Thank you very much. So my second question is more about the more or less last slide you mentioned, where the effect for the most severe symptoms is exactly the opposite of what we would want to expect. And obviously we can't, or we can induce it, but it's ethically problematic, I guess, induce these sedative symptoms. But we can do the exact opposite. So have you ever looked at the development of these ratings throughout the therapy? And is
Mhm.
it the exact reversal, so going up and then slowly going down again? Or how is the shape of this function then? And I mean, this would give us insights if it's
Yeah, yeah.
really the one way or if there is another loop around.
So it's really complicated, that one. The chronicity curve is the best one that I like. The chronicity by itself is a little bit more linear. So we can say the more chronicity people have, the more they show this reduction in reactivity, but their ratings, symptom complaints stay pretty much the same or they get worse, and often they are accompanied by more comorbidity. So as people get older, typically we have now a big sample with OCD and hoarding. They also report more depression symptoms. So there is more comorbidity. There's also more severity overall, where they become more impaired. They go out of the house less. They have less work. They have less friends, that kind of stuff. So that's fairly clear where then that the more you have of that, the less reactive your system becomes. Because a lot of you in your question said, isn't that adaptive? And it is, as I said earlier. You want an adaptive threat system in your life. You want to avoid the snake. You want to avoid bad people. You want to avoid cars in traffic in Münster or Oldenburg or whatever you guys are. You want to ride your bicycle carefully, like these things. You need a fear system, but you don't need that fear system to be active all the time because now you get allostatic load and all that stuff. And eventually, just like the hormone system, which I didn't talk about, that also does that, where then the hormone system doesn't respond anymore. Like these people, if you do a cortisol test on them with high comorbidity, high chronicity, they don't show you a cortisol stress response either. Their cortisol is constantly elevated and there is no response to any specific event anymore.
A lot. Until now, at least I don't see any more questions. Let me have a look at the chat. No. So I
Patrick
would.
has one.
Oh, sorry.
Very sorry.
No,
Um,
that's why we're here.
so you showed like that emotional stimuli will be more tuned, like cells are more tuned for emotional stimuli. Could you
I think,
also show that they have like a higher bias, like in biased competition terms?
yeah, I think so. Yeah.
Okay.
That's exactly what I'm saying. So in terms of biased
Okay.
competition terms, if you make something emotionally relevant for an observer, it will contribute to this thing, whatever that is. It could be a feature, it could be a whole object, it could be part of the visual field that will have a competitive advantage in a biased competition type of deal.
And one second question, where would this bias come from? Like from the thalamus or
Ah,
the
yeah,
amygdala? Like
yeah. Yeah, I
how,
think
and how could you study that, for example?
right. So we tried to study this using concurrent EG fMRI. So if you tag that biased competition, for instance, using SSVPs, oh, where did it go? Sorry, I'm going very fast here. Then you can look where is that bias that you can look at with frequency tagging with SSVPs, where you have multiple things tagged at different frequencies. Now you can measure that bias. Where is that in the concurrent bold? Like where is bold co-varying with the steady state signal that kind of represents this competitive advantage in visual cortex? And the answer to that is kind of here. So it's a lot of frontal structures, subgenual, prefrontal, a lot of parietal things, and also insular things, and basal ganglia, but so far we haven't found thalamus in any of this.
Thank you.
Perfect. That was a great question. In fact, as I said, oh, Julia has a question too. Let me just go back to the
Yes,
end.
thank you. I would be interested if you also had a look up what you in general think about the influence of uncertainty processing and uncertainty
Right.
perception, because there is a lot of evidence that links differences in uncertainty
Yeah.
processing to internalizing symptoms, especially also social anxiety. And I was wondering if that could also interact with these biases towards stimuli.
Right. So that's a funny, it's a great question. It's funny that you bring it up because later today I have a meeting with this person here, Matthias Wieser in Rotterdam, because we look at his clinical data. He has 280 people with these steady state paradigms, and he has the intolerance of uncertainty questionnaire
Yeah,
from each of them.
exactly.
And when we looked, there was not a linear relationship, but there was more this nonlinear relationship along those lines here, where the people who are
Yeah.
really, really super high in terms of uncertainty of intolerance, they just had an overall different pattern entirely, not just the visual stuff, but he also has heart rate, skin, pupil, and all these things. And he also has these environmental assessment things moment by moment. They were also different, but only in those who were very high. Like below a certain
Okay.
threshold, they all looked the same, and other questionnaires were better predictors, like depression questionnaire was a better
Yep.
predictor. But these high intolerance of uncertainty people, they certainly fall into an entirely different category. So
Interesting. Thank you.
similar to this. Yeah.
Yeah.
Great. Any other questions? Because I think we addressed most of your questions, but I don't want to forget them. So here is all the questions that people had. They were amazing. And let me just pick the ones that we hadn't gotten into it. So we already said, yeah, other systems have similar setup. But for instance, the auditory system, lateral inhibition between similar frequencies is not as strong as it is for orientation and visual cortex. So that brings certain consequences. I really like that question, whether it's hard-coded by genetics versus learned. I think a lot of this is learned, and then it prompts epigenetic changes. So in a way, everything that you learn also affects genetic expression. There is now really, really nice data on this, also from rodents, how people used immediate early genes to see how the genetic expression is altered during conditioning, and it is. For those interested in that, Stephen Marin is a good author to look into in the context of extinction and PTSD. And yes, I think eventually people might not think as much about PTSD, but might think of things like, yeah, this person has a hyperactive thread circuit, this person has a lower active, hypoactive thread circuit, or a thread circuit or thread system in the brain that is exhausted, that has high allostatic load and cannot respond to individual events. And in our work, we don't see a whole lot of effects of conscious awareness. So a lot of times we find it difficult to see any differences between the proportion of subjects that are aware and report everything correctly. There is always a subset of them that are unaware and they don't know what's going on, but their physiology, like heart rate, skin conductance, pupil, and also their brain responses often look like the others. So we don't think that awareness is particularly relevant in these very simple conditioning paradigms that we use. But there is Yuri Pavlov, aptly named, a Russian-German researcher, not related to the other Pavlov, but he does a lot of complicated conditioning paradigms where people have to learn many, many different things together, predict the US. And he finds that only in aware people. But in order to be conditioned in his paradigm, you have to learn that only when a tone is played to the right ear and you see a word that denotes an animal and it starts with a P, that is a CS plus. So it's very complicated compared to saying a left shifted Gabor patch is the CS plus. So it might depend on these. There was another great question, the last question, why do some people generalize fear to everything, like in a panic attack, while others keep just very specifically the fear response on the trigger? So that is a great question. I tried to talk a little bit about this at the beginning. when I describe the difference between a dog phobia person and a blood and injection phobia person, you can easily think about a situation where the fear situation is even more general, like in generalized anxiety, where the fear situation is anticipated, imagined future events that could go bad, where people worry a lot about the future and the present and what other people think. And then that in itself just gives rise to more opportunities for generalization, just because the evoking stimulus is less precisely defined. So a lazy, simple explanation that I actually like is depends on the evoking stimulus and whether that in itself is prone to be generalized or not. I think that's the questions. I have to be mindful of your time. So I'm going to leave it right there unless there is any more questions.
I think I would slowly switch over to my slides, but I had one more question that
Perfect.
you maybe could all answer while doing so. Um, so out of all you mentioned, especially the neuroimaging stuff, what do you think is the most promising and also maybe first applicable stuff for the process of diagnostic therapy or even diagnostics, and could be implemented therefore more or less before these 20, 30 years go by, and modeling can be maybe implemented for the use of personal medicine and so on?
Yeah, definitely. It's going to have to be this alpha reduction in extinction, because you can show that the people who keep doing this, where you show them a threat cue and they keep showing heightened alpha reduction in response to that, that is the kind of person who keeps doing extinction learning. And as Lirim said earlier, extinction learning is a model for psychotherapy, or one type of psychotherapy that's rooted in these old 1950s, things, and has been shaped quite a bit. So if we see people do that a lot, normally they are good candidates for exposure therapy, for instance. So we are re-examining that right now. That can be a nice thing to do for treatment matching, where we say, oh, these people go into exposure therapy.
There's one more question from Lirim. So thank you. But there's one more question from Lirim. I've just skipped with the thank you part.
Of course.
Yes, maybe just one jokingly question, I guess. The classic clinical psychology lecture question: why are we not more scared of electrical plugs and things which surround us day in, day out? Do you have a outlook on this day-old question of psychology?
Yeah, you mean like, why are we not more afraid of cars than we are of snakes?
Exactly.
Yeah. So yeah, I think actually this is a good one. The evolutionary preparedness work, like there is older papers by Arne Oehmann, who unfortunately has died, and Susan Meinecker, and a lot of people that try to address that by saying there must be some preparedness, there might be some module. I'm not sure that that is well supported by any data, the fear module idea. So it could still be a cultural thing in my book, but I also note that there's increasingly people who are afraid of cars and who are afraid of electricity, fear of heights, and all these. Fear of heights is different. Forget that I said that. But fear of modern things is definitely on the rise. So it could very well be that some of these prevalence of small animals, prevalence of snakes, prevalence of dogs and other things was still shaped by culture, what people as children picked up from the parents, what is a bad animal from books. I don't know. There is a reason why this is such a nagging question that people like to answer, and I don't really have a great answer. I know that a lot of these things are learned and shaped, but it's also not clear how. Fear of height is a great example where people go to the dentist visits goes horrible, and then they come back with fear of heights. So there is also an unmasking previously extinguished fears that comes back with unspecific stress experience. There is that too. So I have no good answer for that, Lirim. I leave that to you.
Okay, thanks for trying anyways. I always like
Good.
I like to always give this question as a
Oh yeah, no, it's a good one.
okay.
Before I end the presentation, I may contribute maybe a little bit to that question. I mean, of course, it's not identified at all, but I mean, I at least know one or two studies that have been done with monkeys, and they have been ontogenetically isolated. So they've never experienced wildlife. They've been kept in a pretty secure environment where they can control all the stimuli, and they could still show this um, increased fear reaction to snake-like stimuli in comparison to like other things like a weapon where humans usually hunt them, like showing them a bow or so.
Right.
So I would assume that there is something that shaped our system while it developed over time, so phenogenetically. And therefore, we can't really deny this fear
No.
reaction. But yeah, of course, we don't know the full detail. But I thought this studies were very impressive.
hmm No, I agree. Yeah, no, this is fun to read. Yeah, there's old work by Mortimer Mishkin, and others that you can read. That is really interesting. Also, when they took out the amygdala or the entire medial temporal lobe of those same monkeys, it's interesting. Great point, Sebastian.
Then thank you, and thank all of you who've still been there and who have been there, maybe watching this on YouTube. I hope you enjoyed. We did. And yeah, so, and if you especially would like to have a more detailed look in Andreas' work, we linked all of his more or less relevant socials and also websites where you can look up his either his personal postings on Blue Sky and also Slab stuff, his papers and publications. And we will also, as always, share these slides. Might take a little longer due to the exams, but yeah. With all of this being said, I can only thank you again, and have a great evening. Enjoy, and good luck with everybody who's writing exams in the next weeks, month.
Good luck, everyone. It was a pleasure. Thanks for the great questions, and have a good night.
Thank you. Have a great day. Bye bye.
Bye.